Okay, in this video we're going to be talking about hybridizations. So hybridizations is when you are using DNA or RNA probes to detect DNA or RNA within a gel, okay, or what we call a blot, or within cells or an organism. And you can do the same thing with proteins, but of course to detect the proteins you can't use RNA or DNA probes. You need to use what we need to use antibodies. Okay, so nucleic acid hybridization depends upon the fact that if you have two sources of RNA or two sources of DNA or a source of RNA and a source of DNA, if you have two polynucleotide strands that are reverse complementary to one another, regardless of where they come from, they will hybridize together. Okay, so regardless of where this sample of polynucleotides come from, regardless of where this sample comes from, if you put them together and they're reverse complementary to each other, they will form duplexes. Now that duplex could be DNA-DNA, it could be RNA-RNA, and it could be DNA-RNA. In order to detect a specific fragment within a gel or a specific DNA within an RNA within an organism, what we need to do first is make a probe. Now that probe is going to be a small piece of DNA or a small piece of RNA that is reverse complementary to some part of the DNA that you're looking for, or some part of the RNA that you're looking for. And when we make that probe, we have to add something, some modification to that probe, something that we can detect. So when you make a probe, the nucleotides that you use to make it are modified in some way. The most old-fashioned way to do this is to make the probe using nucleotides that have been made with radioactive phosphorus, so P32. And that way, since it's radioactive, it gives off high energy particles that can be detected via autoradiography. Since it's radiation, you need to have like a big old plastic shield between you and the samples that you're working with, and it's not the safest thing in the world, and it's not being used quite as much. Nowadays, we label these probes with nucleotides that are bound to things that we can detect in different ways other than radiation. So fluorescence, for example, or enzyme conjugated antibodies. So ways that we can detect these things more safely. Now, you don't need to know the details about these detection mechanisms. You just simply need to know that we're making probes using nucleotides that have been modified in such a way that we can identify where those probes are within a cell or on a blot. Okay, so let's talk about blots then. We've mentioned this word a few times. What is it? Well, a blot is this nitrocellulose or this nylon membrane that you place over a gel, and all that DNA or all that RNA is then transferred from the gel onto that nylon membrane, and that nylon membrane then we call a blot. So the first step of making a blot is to run a gel. So you need to run a DNA gel, or you need to run a formaldehyde gel for RNA, or you need to run a protein gel, an SDS-PAGE gel. Now, how you actually detect the proteins is going to be different than DNA and RNA, but this first step is the same. You've got to have a sample of DNA, RNA, protein. You have to load them onto a gel, and you have to run the gel. Next, you have to prepare the blot. So here's that gel, whether it is a protein gel or a DNA gel or an RNA gel. We've got that gel. And we place that gel onto a sponge in a solution of some sort of buffer. Then on top of the gel, we place a nitrocellulose blot, okay, or more commonly a nylon blot. It's a little bit stronger material. And then on top of that, we place a whole bunch of paper towels, and then on top of that, we put some sort of heavy weight. And what's going to happen is the buffer is going to be wicked up through the sponge, through the gel, into the paper towels. And as it is wicked up, it carries along with it, with that buffer, it carries along all of this protein or DNA or RNA. And that protein or DNA and RNA then gets transferred from the gel onto the blot in exactly the same position as it was in the gel. But it's invisible. We can't see anything. So when you're done with a blot, when you pull off these paper towels, when you pull off that book, when you pull off everything, that blot is you can't see anything on it. It's just a white piece of paper. Blots that are made with DNA are called southerns. Blots that are made with RNA are called northerns. And I forgot to add this, and we'll see it in a few slides. Blots that are made with proteins are called westerns. All right, now the first person who invented these, his last name was Southern, and then Northern and Western were just sort of jokes after that. Step 3. Now we hybridize that blot with a probe. For northerns and southerns, it's the same thing. You're going to use either DNA or RNA that has been labeled in some way. For westerns, it's going to be a little bit different. We're going to use antibodies. We'll talk about that in a minute. So here's an actual experiment that was done using a northern blot to ask a question, to sort of test a hypothesis. And the hypothesis is that these leaves are mutant leaves. They have this mutation called stay-green. Normally, these leaves would eventually senesce and turn yellow, but these don't. And the idea is the hypothesis was the reason is because these leaves lack a particular messenger RNA that encodes an enzyme that breaks down chlorophyll. Okay? So what their hypothesis is they should see that messenger RNA here in the wild type leaves, but not here in these mutant leaves. So they purify RNA from both sets of leaves, wild type and what are called stay green. They run them out on a gel, a formaldehyde gel. They make a northern blot and they hybridize it with the messenger RNA that they think the yellow leaves have, but the green leaves lack. Right? So there's going to be lots and lots and lots of RNA in each one of these lanes, right? All of the RNA that's expressed in all of these cells. Okay? Lots and lots of RNA. But we are probing for a very specific RNA. And you can see in the wild type, which is lane 1 and lane 3, you got lots of that RNA, lots of that RNA. But take a look in lane 2 and 4 that represent RNA that was purified from these stay green, you don't see that RNA. So this supports their hypothesis that something is wrong with these particular mutant cells, leaves, that they don't produce the specific messenger RNA, and then that's the reason why they have this mutant phenotype. Well, let's talk about protein blots. So protein blots are called Western blots, but maybe more commonly nowadays simply immunoblots. And certainly in the paper that you guys are reading, that's what they refer to these as. Now, although the blot itself is made exactly the same way as a northern and as a southern, the way that you probe a Western blot has to be different. Proteins are inherently different than nucleic acids. And the way that we probe Western blots is by using antibodies, because antibodies bind to proteins. And you have to do it twice. Okay, you first have to use what's called a primary antibody. That's the one that detects your protein of interest. That's the one that's going to be sort of difficult to get. You either have to send your protein of interest to a company who will make an antibody for it, or you convert your protein into a fusion protein that's tagged with something that you already have an antibody for. For example, hemagglutinin. Right? So instead of having to make an antibody specific to your protein, you modify part of your protein so that it binds an antibody you already have. Okay, that's why these HA tagged proteins are really important. Okay, so first you hybridize your blot with your primary antibody. And then you probe it again with a secondary antibody that recognizes primary antibodies. They recognize just antibodies in general. In addition, these secondary antibodies, they have attached to them something that is detectable. Maybe a fluorescent dye or an enzyme that catalyzes a chromogenic reaction, something that you can detect. And so the way a Western blot or an immunoblot would work is, here is your blot. Here is the protein on the blot, the protein of interest. Remember, that blot's going to have a whole lot of other proteins on it. You first incubate that blot with the primary antibody. Then you incubate that blot with a secondary antibody that is modified to have some sort of detectable function. And then you wash it, and then you add whatever it is you need to sort of get this color change. So adding substrate to develop color, you're developing that blot. Now, the journal article figure 1E and lots of the other figures within that journal article uses immunoblots, uses Western. So if we read very carefully the figure legend for figure 1E, it says immunoblot using lysates from HeLa cells transfected with control, lane 1, PCMVA-HA-JM-JD3, lane 2, or PCMV-HA-JM-JD3, lane 3. Now, we've already discussed what these mean. JM-JD3, this PCMV-HA-JM-JD3 is this entire construct, expression vector construct, that they have cloned in the cDNA that encodes the protein of interest, JM-JD3, that will then be tagged by HA, hemagglutinin. So now that fusion protein that gets made is going to look like this, JM-JD3 protein attached to hemagglutinin. What is making this protein? Where are these cells being transfected? They're being where are these construct, this vector being transfected? They're being transfected into HeLa cells. These are human cells. They are derived from a woman who died in 1951. Her name was Henrietta Lacks, and she had cervical cancer. Before she died, biopsy was taken of those cancer cells. Because they were cancerous, it's very easy to keep them dividing and dividing and dividing. And even to this day, scientists study HeLa cells. They're a good way, easy way to study proteins in the context of human cells. So the researchers transfected this expression vector into HeLa cells, forcing those HeLa cells to make all sorts of proteins, right? These HeLa cells they're just a living cell. So they're making lots and lots of proteins, but now they're also making this fusion protein here. And the researchers want to prove to you, the reader, that yes, indeed, these HeLa cells are making our fusion protein. So they lysate means they break open those HeLa cells. They take all the protein from those HeLa cells, they run them out onto a gel. Okay, so there's lots and lots of proteins, not just this one, but lots and lots of other proteins. And then they make an immunoblot, and they probe the immunoblot with an antibody that specifically detects this HA portion of the fusion protein. They then develop it, and this is what they see. Now, that's our negative control. They don't expect to see anything because they did not transfect those cells with a plasmid. They were empty cells. Okay, so the only proteins those cells made were their endogenous proteins. They certainly didn't make any cells, any proteins that had this HA tag to them, and so that's why we see nothing in lane 1. In lane 2, it is the HA-JM-JD3, so the normal cDNA that encodes the normal JM-JD3 protein. And although they get these two weird bands here that they don't really explain, it's this one here that they say this is our full-length protein. So they're proving that in this cell type, it does indeed make our fusion protein. And then in a third set of cells that they transfected it with JM-JD3S. It's a little mutant version of the protein. It's a smaller version. That's what S means, of that protein. They then transfected those cells with that expression vector. They burst them open, and they loaded them in lane 3. Again, incubated with the antibody that detects the HA. Right, all of these are on the same gel, and sure enough, look at all that protein. So those particular cells are making lots and lots of this JM-JD3S protein. Okay, so the authors then have done this experiment in which they're forcing HeLa cells to express this fusion protein. Now, that's just setting up a tool. Okay, the question that they're actually asking, we haven't gotten to yet. I'm going to make a video specifically for that a little bit later. Okay, blots are a way to visualize proteins, RNA, and DNA in the context of a gel. Okay, in the context of a blot that you make from a gel that you have already run. In situ hybridizations and immunohistochemistry is a way of detecting RNA and proteins within cells themselves. Right? You don't burst open those cells and grab the RNA out of it or grab the protein out of it. You look at it within the cells itself, and those cells can be just single-celled or they can be part of an organism. In situ hybridizations reveal RNA expression in cells. And so your probe is going to be either DNA or RNA. And the target is going to be that complementary RNA within the cells of a tissue or the cells of an organism. So that, for example, this is a Drosophila embryo that has been probed with a probe that detects a RNA called Fushitarazu. And you can see that Fushitarazu is expressed in a very specific subset of cells within that embryo. It's pretty amazing, pretty striking. In fact, Fushitarazu is Japanese for every other stripe. Now, here's another example where we're looking at a zebrafish embryo and we are probing with a probe that detects RAG1 messenger RNA. In this one here, it shows up very specifically here in the thymus glands. This is a mutant that doesn't have thymus glands, and sure enough, because of that, it doesn't have that RAG1 expression. So this is a useful way of detecting a mutant from a wild-type embryo. So these are called in situ hybridizations. They are a way to reveal where RNA is expressed within cells or within organisms. Immunohistochemistry, histology, tissues, organisms, right? That's what the histo is there. Immunohistochemistry is like the same thing as in situ hybridizations, but yet it reveals proteins in cells. And figure 1b and many of the other figures within the paper take advantage of using immunohistochemistry. So if we look at the figure legend of figure 1b, it says HeLa cells transfected with PCMV-HA-JM-JD3, and then white arrowheads indicate transfected cells. So we're going to concentrate right here on the right-hand images. This is that same thing. This should be familiar to you. This is the expression vector that ends up generating a fusion protein, our JM-JD3 fusion protein fused to hemagglutinin. Now, instead of taking these HeLa cells and bursting them open and making lysates of them and running them on a gel, we're taking those cells and we're incubating them in the presence of the antibody, directly incubating them in the presence of our anti-HA antibody. Okay, in order to do that, we have to kill the cells off. Okay, we have to kill them off. We have to permeabilize them so those antibodies can get into the cells. So the primary antibody goes in, and wherever that JM-JD3 protein is, it binds to that HA. Then we add our secondary antibody, and that secondary antibody is conjugated to an enzyme that gives rise to a fluorescent that fluoresces. In fact, it fluoresces green under the right conditions. But again, the cells are dead at this point. So what we're seeing here is 1, 2, 3, 4 cells, each one of these expressing the protein. Now, I should take advantage of showing you this left illustration. This left illustration shows you all of the cells that are visible under the microscope. DAPI stains the nucleus. Therefore, it stains all of the cells. Notice only a subset of these cells, of the cells that are visual under the that you can see under the microscope, are actually transfected. Right? This one here, it's visible here, but it's not showing green. This one here, it's visible here, but it's not showing green. So not all of the cells were successfully transfected, and we know that that's the case, that transfection is a fairly rare process. Okay, here's a summary then of blotting. You can run DNA, RNA, protein onto a gel. You can take that gel and make a blot of it, so that nitrocellulose membrane. If it is DNA, we call it a southern blot. If it's RNA, we call it a northern blot. And if it's protein, we call it a western blot, or an otherwise called immunoblot. Now, to probe that blot, if it's a DNA or RNA, if it's a southern or northern, we can use single-stranded DNA or RNA. If we're going to probe an immunoblot, we have to use an antibody. We have a primary antibody and then a secondary antibody.